So this group I told you before that it's one of the largest subgroup of the proteobacteria, and this group is actually divided into many orders, but the prominent orders, or you can say the dominant orders, or the members which are displaying some dominant activities. So we'll discuss those orders, right? So there are some prominent orders, including pseudomonadales, as its name indicate. How how do you know that these are the orders? Because of the suffix, right? Ales represents the order, right? So there are five important orders that we will discuss in this course, which are the pseudomonadales, legionales, vibrionales, enterobacteriales, and the pasteurellales. So pseudomonadales, because its name indicate that one of the most important genus of this group is the pseudomonas. So pseudomonas is known for many differentiating properties, and again they are gram-negative rods or cocci, and they are aerobic in nature. So an important genus is, I told you, the pseudomonas, and they are motile. So motility is because of a single flagella, or maybe because of a lot of flagella, tuft of flagella, right, which are present at the poles, at the poles, not on the entire bacterial surface, right? So they are very common because they inhabit almost all the natural environments. So very common in soil and the other natural environments. And monas is well known for its pigment production, because they produce the pigments which are not intracellularly located, rather they excrete the pigments. They excrete the pigments extracellularly, and these pigments, they are water soluble in nature, and whenever they are released and you grow the pseudomonas species uh, in the agar plates, what happens, they release their pigments, and these pigments, they diffuse, and whenever they diffuse, you start noticing the uh, colored colonies, or you can say in the colored area where the growth is present. So pseudomonas is very much uh, eye capturing, I must say, that you can easily differentiate pseudomonas growing in any plate, right? As you can see that this is the color of the pigment. And one of the most prominent member that you have already heard is the Pseudomonas aeruginosa, and it also produces a blue-green pigment, which is shown in the picture here. As you can see that this picture is actually a picture of antibiotic sensitivity. So wherever any antibiotic is placed and Pseudomonas is sensitive, so you are not able to see its growth, and you will find a clear region, and in that clear region, uh, since no growth is there, so there is colorless region, right, where all the other sections of the plate, uh, they got a color pigment, and it is visible in blue green type of coloration. Any other member which is important in this regard? Pseudomonas aeruginosa is well studied, or you can say the type strain of this is group Pseudomonadales, right? So well characterized. Another important member is Pseudomonas syringae, and this is one of the plant pathogen which is known to cause plant infection, particularly in different rice species. So importantly, this Pseudomonas is very much important regarding its ability to produce unusually large number of enzymes, and it is also able to metabolize a wide variety of substrate. Okay. And secondly, since I told you before that once any organism is capable of degrading a wide range of uh, complex compounds. So definitely it can survive in different nutritionally low environments. So definitely they are also a threat for the hospital environment and in the pharmaceutical industries, like in the same way we discussed in the Burkholderia species. But definitely they are much more abundant than the Burkholderia. And the second threat is that they are showing a great resistance to antibiotics, and mostly they have this mechanism that they have cell wall porins, so they need to excrete out. They, once they are treated with antibiotics, they used to excrete those antibiotics out of the cell because of this presence of the cell wall porins. So this is how they display their resistance towards the antibiotics. Another important thing is they can grow at refrigerator temperatures, and definitely they can also, I told you that they can utilize many organic compounds including proteins, lipids. So definitely they are also contributing, they may contribute very easily into different food spoilages as well, right? Now another group is in the order Pseudomonadales. So one of the member we discussed about Pseudomonas. Now some other members in the group Pseudomonadales are Azotobacter and Azomonas. So they are also free living soil bacteria, and they're helpful in the nitrogen fixation, but they live freely. They do not form any contact or any symbiotic relationship with the plants. So, and their characteristic capsule is present, and because of that capsule, heavy capsule, their morphology becomes a large ovoid cell, right? So definitely we discuss in these two because they are participating definitely in the nitrogen cycle in the world. Another important member of this Pseudomonadales group is uh, Acinetobacter. Now this Acinetobacter is well known for its ability to resist antibiotics, right? And it's one of the member is the Acinetobacter baumannii, and this organism is actually displaying a great deal of antibiotic resistance. And can you imagine that actually these pathogens, they target those individuals which are the hospital patients and their immunity is compromised. And secondly, when the immunity is compromised, so second thing is that they are uh, they are showing extreme resistance to different antibiotics, right? So their infection is hard to treat because they are showing the great deal of resistance against antibiotics, right? So it has unusual high mortality rate in the hospital acquired infections, you can say, or in the patients which have immunosuppression, or you can say which have a compromised immune system, right? So this is a very common problem in the hospital environment, Acinetobacter. And as we discussed that it is once established in a hospital, it's difficult to eliminate. I told you the reason that you usually used to eliminate the infections by treating them with antibiotics, but they are mostly showing the resistance towards all the antibiotics. Legionales is another order, and the important genera includes Legionella and Klebsiella. So these two genera, they are important genera in the order Legionales in the gamma proteobacteria. So Legionella is important because it was originally isolated for causing a form of pneumonia, which is also known as legionellosis, right? So they colonize such habitats as warm water supply lines in the hospital and water in the cooling towers of air conditioning systems. So they may colonize these things, and these water supplies and the cooling towers of air condition, they may be a source of these bacteria, right? So they can survive and they can reproduce within aquatic amoeba as well, right? So that's why it is difficult to eradicate them in water systems, because they hide themselves in the another organism, right, which is the mostly aquatic amoeba. So water system में इनको eradicate करना थोड़ा सा मुश्किल हो जाता है, because they can survive and they can also reproduce within the other organism, right? So another thing or important genus is Klebsiella. So you must have heard about Klebsiella burnettii, which is responsible for causing Q fever, right? The spores, they are one of the most resistant spore regarding their uh, temperature sustainability or stability, right? So the thing is that Klebsiella requires a host to reproduce, and it is mostly transmitted by aerosols or the contaminated milk. So that's why milk is heated or pasteurized until the temperature at which the spores of these Klebsiella burnettii, they are killed, right? So they form, this is unique in that sense. Usually gram negative organism, they do not form any spore like body, but Klebsiella, they form a spore like body which is present in the Klebsiella burnettii, and it's also shown in the picture as well, right? And this is very resistant to the stresses of airborne transmission and the heat treatment, right? So Klebsiella is unique in that sense, that it is displaying that property which is absent in almost all the gram negative organisms, right? So this was the order Legionales, and we discussed two members, Legionella and Klebsiella. Now moving towards Vibrionales, and you have heard about Vibrio, but I don't think so much in detail. But and they are the gram-negative organisms which are anaerobic in nature, and as its name indicates that they are slightly curved, and many of the members, they are slightly curved. Right. What about the others which are not included in that many? Why they are included in the Vibrionales? Because I told you that these classification schemes, they are not now built on the morphological properties, but they are built on their 16S ribosomal RNA analysis. They are mostly found in aquatic habitats, and one of the member is important member is Vibrio cholera, and it is well known for the spread of cholera, and it is cholera is what it is simply watery diarrhea, and mostly it targets it is you can say cholera. It is I must say its life span is short, and once initiated, its infection is initiated, it's usually completes its life cycle and you suffer the symptoms, and then everything it gets normal and it gets clear from your body. But for for very few individual it gets life threatening if the loss of minerals and loss of water is not covered by the different supplementations. So it's usually life threatening for the infants, I must say. Another important pathogenic strain is the Vibrio parahemolyticus, and it also less serious form of gastroenteritis or a gastric uh, infection or sort of food poisoning, and it is uh, usually transmitted by eating the uncooked or raw cooked shellfish. So basically, Vibrio parahemolyticus, it actually inhabits the shellfishes. So if the seafood is consumed undercooked, or you can say in raw condition, so you may get the form of gastroenteritis caused by Vibrio parahemolyticus. The order Enterobacteriales, it comprises of the members which are anaerobic, but the nature is facultative. So they are facultatively anaerobic and obviously gram negatives. But the common group properties include their motility, that if they are motile, then the flagella is not polar, rather it is peritrichous, means distributed all over the surface, and they are morphologically rods, and there is among enterobacterials, it's a very large group, but one of the most important group includes the enterics. And the enterics, actually, the word enterics, it refers to the intestine, and most of them are the natural inhabitants of the intestine. So most of them, they are the normal inhabitants. So they inhabit your gut, and they are the gut members. So they are the fermenters of glucose and the other carbohydrates, and they have a special organelle for, I must say, for the adhesion to the surfaces or the mucous membranes, right? So this is known as fimbriae, or you can also say pili. So there are different types of pili which are present, and one of the pili is used for adhesion, and the other specialized types include sex pili, or you can also say F pilus. You have heard about it in the previous semesters, and the F pili or the sex pili, its role is in the exchange of genetic material between the cells. So, and the genetic material, whenever it's exchanged, so the information is also exchanged, which often results in the spread of the antibiotic resistance, right? So I think the mechanism, that how the things they are transmitted through this genetic exchange, is, I think, well explained in the previous semester, so I'm not going into the details. So these are some common properties. One more thing, that many members of this group, and they are specialized, or they produce special kind of proteins which are known as bacteriocins. And these bacteriocins, they are the basically peptides that are called, as you can say, inhibitory peptides, or they used to inhibit or kill the other cells, uh, the closely related cells, other closely related species. So the mechanism to produce bacteriocin is to reduce the competition between the members of closely related species. So they also produce different bacteriocins, right? So one of the most common and well-known member is Escherichia, and you are well aware about this organism because so far this is the most common organism you have dealt with, and it is also one of the most common inhabitants of intestinal tract, and it's, you can say, a sort of a pet microorganism in the field of microbiology. So the most familiar and the most studied or well studied organism, bacterial species. So it's important in a way that generally it's not the most of the strains of Escherichia, it's a genus. Escherichia is a genus, so most of its strains or species, they are non-pathogenic and they are the normal inhabitants. But since they are normal inhabitants of your gut, so they are the part of the fecal material as well. So their indication in the water or its indication in the food, it is representative of the fecal contamination, because they are present naturally in the fecal material. So if their presence is detected in water or food, so it indicates that your food or water is contaminated with the fecal material. So I told you that it is not pathogenic, but it may cause urinary tract infections in case if the immunity is suppressed, and it may act as an opportunistic pathogen in that case. But naturally or generally it's not pathogenic. However, there are some strains of E. coli that can cause severe form of diarrhea, and that could be life threatening. So some of those strains are also exist, which are pathogenic, and they produce toxins, and these toxins basically they damage the linings of the membrane of you can say intestine. So they may cause diarrhea and very serious foodborne diseases. And one of the example is this species strain. And now, since it is the well-studied organism, so I can give you the example of how the things they are written when the classification is explained, you can say. So while taking the example of E. coli, you can see that its domain is bacteria, its kingdom is bacteria, and phylum we are studying proteobacteria. It belongs to the class Gamma proteobacteria. We know its order is Enterobacteriales, and its family is Enterobacteriaceae, and the genus is Escherichia, and the species is Escherichia coli. So that Salmonella is another well-known genus, and you are well aware about Salmonella, and they are also the common inhabitants of intestine or GIT of many animals, especially poultry and cattle. So in case of poultry and cattle, they are the common inhabitants of GIT.
other than the E. coli and other members of Enterobacteriaceae, and they may contaminate the food under unsanitary conditions, or since they are also present in the intestinal tract, so definitely the source of their contamination would also be the water and food which is contaminated with the fecal material. So mostly mm, they are responsible for causing infection in the warm-blooded animals, and they those which cause infection to warm-blooded animals, they are considered as a single species, and which is known as Salmonella enterica. And this species then further divided into 2400 serovars. And I told you that what is serovars means that these serovars, they differ in their serology, means the components of the cell walls and the reaction to reaction of your immune system to these serotypes. So they are different in terms of their surface chemistry. Right. Another important thing is that although they are mostly targeting towards warm-blooded organisms, but one of the member that is known as Salmonella bongori is unique in that sense that it is the resident of a cold-blooded animal, and it was originally isolated from lizards, and lizard is one of the cold-blooded animals, right? So Salmonella is responsible for causing a well-known type of fever, which is known as typhoid fever, and it's caused by Salmonella typhi, and it is the most severe illness caused by any member of the genus Salmonella, and usually typhoid is very much severe if it is targeting the infants and the children. So it may be a worst case. So it is very severe form of illness by any member of the genus Salmonella. Another disease or GIT infection is the salmonellosis, and this salmonellosis is one of the common form of foodborne illness, and definitely this is transmitted through different foods like eggs and chickens and the undercooked meat, right? And now moving towards Shigella, another well-known organism. So Shigella is responsible for for a disease known as dysentery, or it is also known as shigellosis. So dysentery can be caused by Shigella, which is also known as bacillary dysentery. A type of dysentery is amoebic dysentery, which is caused by ant amoeba histolytica, but the cause is an amoeba. But here in this case, the dysentery is the bacillary dysentery, which is caused by the Shigella species. So this is also known as shigellosis, and you can see an image here, and it is showing a picture of SS agar, and this SS agar is representing the two different types of colonies, and the same medium can be used for the differential colonies of Salmonella and Shigella. So this is a differential medium, means on a single medium you can differentiate the two types, Salmonella and the Shigella, right? Because under microscope they look like same, they mostly share the biochemical reactions. So a quick test is that you can plate the organisms on this agar. So you can see that Salmonella, it gives black color colonies, and Shigella on the SS agar, it gives yellow color colonies. So it gives a quick differentiation between the two things. So somehow if the two cultures they get mixed, then this is an easy way or quick way to separate the two and purify them. So they are found only in humans. So Shigella can cause dysentery, and the different strains of Shigella can cause dysentery of different severity, but some strains they can cause the life-threatening dysentery. Right. Similarly, we can discuss Klebsiella. It's also the member of Enterobacteriaceae, and the family is Enterobacteriaceae. And Klebsiella is commonly present in soil and water, and definitely the source is um, intestine, and they are also naturally present in soil and water, and they are capable of fixing nitrogen. So this is one of the unique feature of Klebsiella among enterics, and that it has a nutritional advantage in the populations where low content of nitrogen is available, right? Or it can survive in that environment where the populations they have poor nitrogen content in their diet, right? So since they can fix also the nitrogen from the environment, so they can easily survive. So one of the member is responsible for causing pneumonia, which is a serious form of pneumonia in humans. And definitely it causes in those patients which have immunocompromised system, so immunity is low. So that species is Klebsiella pneumoniae, right? Klebsiella under microscope is visible with its thick uh, capsule, as you can see. So because of that capsule and the colonies of Klebsiella, they are slimy and they are the large colonies. Another member is Serratia, and Serratia is well known for its pigment. So it generates a red color pigment. So under hospital situation, and this organism can be found on catheters and in saline solutions or in other supposedly sterile solutions, and it may contaminate these things, so that it can be detected by its production of red pigment. So another member is so such contamination is responsible or may cause different urinary and respiratory tract infections in hospitals. So another genus is Proteus. Proteus is well known for its differential motility pattern in agar plates. So it has a swarming top type of growth. So the pattern is visible here, that it forms in it forms a sort of rings, and these rings they indicate the different motility pattern. These type of rings they are also known as concentric rings because they share a common center. So these rings form because newly formed Proteus species or newly formed Proteus cells, they are highly motile by peritrichous flagella, and they start moving outward from the place and they are originally present, and once they are mature they lose their flagella. and then they again divide. So this results in this form of rings, right? So for example, the cells here, after just formation, they will start moving or swimming or swimming uh, outwards, and they will start moving out from all the directions, and once they get mature, they stop their movement, right? And they start preparing themselves for division, right? So this forms a ring, right? So a new generation will again be generated, and they will start moving outward, and again after getting mature they will lose their movement, so they'll stop moving then. So this is how this swarming type of growth is visible, right? So this organism has this distinctive colonial feature, but along with that it is very important in different urinary tract infections and the infections in wounds. One of the well-studied species is Proteus mirabilis, and then we have Yersinia. Yersinia is well known for an historic infection which is known as plague. So it is responsible for causing an infection which is also known as the Black Death of medieval Europe. And plague is very much historical, and you can search the details over internet. And Yersinia pestis is that strain which is responsible for that historical plague event, and it actually inhabits rats and squirrels, and they carry these bacteria, and they transmit the organisms to human, from animals to humans. So it's one of zoonotic infections, and and the transmission is human to human transmission is airborne, right? So that's why it was very severe, and that's why its spread was so quick. Another member is Erwinia. Erwinia usually targets plants, and so they are the plant pathogens, and they cause some serious plant diseases. So how they generate the pathological symptoms? So they produce enzymes, and those enzymes, they hydrolyze the pectin between the individual plant cells, so it results in the loss of the strength, or you can say it results in the plant cells detachment, and it leaves a softening effect, and this term is known as plant rot. So they are responsible for causing different types of rots in plants. So another member is Enterobacter, and you have heard about it in, I think, in your lab classes, or you can say in your lab sessions while doing MVIC test. So MVIC test is the key test to differentiate between E. coli and Enterobacter. So some common species of Enterobacter includes Enterobacter cloaca, Enterobacter aerogenes, and they cause UTI and hospital-acquired infections. So they can be easily differentiated from E. coli through MVIC test. And this was all about the order Enterobacteriales, and another order is Pasteurellales. So they are usually non-motile, and they are best known as human and animal pathogens. So they mostly target the animals, and the genus Pasteurella within this order is well known for the as a pathogens of domestic animals, and They cause sepsis in cattle, fowl, cholera in chickens, and the other types of fowls, and pneumonia in the different types of animals. And it's well-known species, or you can say the wild type species, Pasteurella multocida, and it can be transmitted to humans by dog and cat bites. So another member is Haemophilus. It's also well known, but its name is misnomer. It's very important genus of pathogenic bacteria, and they usually inhabit mucous membrane of upper respiratory tract, mouth, vagina, and intestinal tract. And the best known is in Haemophilus influenzae, and it was erroneously believed that it is responsible for causing influenza, but generally it is not responsible for causing influenza, but it forms a sort of pneumonia type illness, and its name is because of the reason that why it is called as hemoplast, because it is unable to synthesize an important part of cytochrome systems, which are responsible for taking part in the respiratory reactions. So definitely they obtain these substances from the host and from the heme fraction of the host, which is known as X factor of the blood hemoglobin. So that's why, since they require heme for their growth, so they are known as, or they are named as hemoplast. So heme loving, you can see. So that was all about proteobacteria, non-proteobacteria gram-negative uh, organisms. So they are distinctive in that sense that they are photosynthesizing bacteria, and they are morphologically different from the other proteobacteria. So they may be called as non-proteobacteria, which are grouped in the volume 2, right? So these organisms, they are mostly photosynthetic, and there are some of the characteristics which are summarized in this table. And one of the common member that you have, I think you know it, is the cyanobacteria. So one of the main group of these non-proteobacteria, but which are also gram-negative, and they are the part of the volume 2, which is also known as the proteobacteria. So it is the cyanobacteria, and its example is cyanobacteria and abina. So it is, though you can see in this table, these are those groups. So there are 5 different groups, and you can see it includes cyanobacteria, it includes green non-sulfur bacteria, green sulfur bacteria, purple non-sulfur bacteria, and purple sulfur bacteria. So the name indicates the presence of different pigments and the ability to use sulfur as electron donor or not, right? So this is how these groups are classified. So, and they are also classified according to their ability of producing oxygen during photosynthesis. So if they are able to produce oxygen during photosynthesis, so they are oxygenic, and the rest are anoxygenic, which do not generate oxygen. So as you can see that among these 5 groups, only the cyanobacteria, the group cyanobacteria is only group which is oxygenic in nature, and they carry out photosynthesis just like plants, and they use water as a electron donor. So this photosynthetic reaction is same as that of plant, that whenever carbon dioxide it reacts with water, it generates oxygen and energy and carbohydrates, right? But these organisms, the other four organisms, so these out of these four organisms, instead of using H2O, they use H2S for uh, carbon dioxide reduction. Means they combine carbon dioxide not with H2O, but rather they use H2S for the reduction of carbon dioxide. And these H, so definitely they should live in those environments which are rich in the sulfur, or which are rich in the sulfur compounds, right? So two of the groups, including green sulfur bacteria and the purple sulfur bacteria, they are usually those groups which use H2S for carbon dioxide reduction. And definitely whenever they use H2S, so they do not release oxygen, rather they release sulfur, and that sulfur is actually deposited in the form of granules inside the cells. So they deposit the sulfur granules inside the cells. So the other organisms include green non-sulfur bacteria and the purple non-sulfur bacteria. They use different organic compounds, whatever available in their inhabiting environment. So they use different organic compounds as a source of electron for carbon dioxide reduction, and definitely they are also an oxygen.